Okay, so we are done with the basic packaging of the DNA into our previous class, and right now we are moving on to the next topic, which is 6.2, the search of the genetic material. The search of the genetic material that people did not know. Into the 1925, let us say that if you walk up to a scientist and you ask him that according to you what could be the genetic material, then the scientist must have answered you that most probably it is protein. Now your next question would be that why? The number of amino acids that are making the protein are 20 as compared to the number of nitrogenous base pairs that bring about the variability into the DNA are somewhat around 4 or 5. So it makes sense that if you have got more amount of monomer, different kinds of monomer as in protein, which are 20, so you can bring about more number of changes, and the inheritance is all about variation into the population. So people, like, by default they were assuming that if someone is able to explain that which will be the genetic material, then it will turn out to be the protein. But this was wrong, that with subsequent experiment that we are about to see. Okay, so now let us start with the very first experiment, which is the transforming principle experiment by Frederick Griffith. So the year was 1928, and into the 1928 he was working with the bacteria, and the name of the bacteria was Streptococcus pneumonia. So the name of the bacteria was Streptococcus pneumonia. So as you can see right here, that this bacteria was coming into two strains. So strain 1, and although the name was strain S, and the another one was R strain. Okay, what do we mean by that? That how did they get their name strain S and strain R? So if this is the bacteria, normal bacteria, if this is genetic material, it does not have outside of the plasma membrane, it does not have anything. That is why it has got somewhat a rough kind of appearance. This is rough relative to the S strain, which was very much smooth and shiny in its outer appearance. So if this is the S strain of the same bacteria, the genetic material, and along with the genetic material, it had one very nice covering out of it, and this covering was our glycoprotein layer, and this was virulent in nature. Okay, so this was the reason why people were getting those diseases. So this outer covering will be the let's say glycoprotein. Glycoprotein. So he set up one particular experiment. In this one particular experiment, what did he do? So he took mice. So this was the mice, and he is going to incubate this particular mice with a smooth strain. So we had this smooth strain, and since a smooth strain was virulent, so it had this covering outside of it. So that after some time, what he observed, the bacteria or the mice died. Now he's going to do it with R strain. So let us see that what is going to happen with the R strain. If this is the mice and he used this normal R strain, the bacteria lived. It did not do anything. Okay, it was able to survive very nicely. So we were very much sure that this outer covering was virulent in nature. Okay, so virulent means that it was pathogenic. Okay, it was pathogenic. It was able to cause disease. Now here comes some interesting setups. So what he do that he took the bacteria and he made sure that if the bacteria is dead, okay, so he took the the smooth bacteria, the smooth strain, that outside of which we had this glossy covering, but just before it, okay, just before incubating it, like injecting inside the mice, what did he do? He heat killed it. And while he was heat killing it, he denatured the bacteria, isn't it? If this is the he heat killed it, he denatured the bacteria, and now the integrity of this glycoprotein is not there anymore. So that is why the mice survived. Okay, so what result did we see? That mice actually survived because now those glycoprotein which were very virulent, now it is not there anymore because heat killed it. Now this is the most interesting setup. He had this living mouse, and along with this living mouse, he had all those bits and pieces of heat killed bacteria that he used into above experiment. So let us say that all those bits and pieces, that all those macromolecules and bits and pieces he had, because this was heat killed bacteria, and along with it he added a rough strain. So this was the rough strain. Now the bacteria died, the mice died, and this was a very, very, very interesting observation. What did he conclude? That there was something, okay, let us say this was that chemical X, okay, so let us say this was the chemical X, which actually somehow transformed this particular R strain into S strain. Okay, so he was able to convert into S strain, and that is why the mice died. So this was the very first experiment that Frederick Griffith did, and he wanted to say that indeed that the information, the genetic information is going from one bacterium to another bacterium. That means that it has to be the transforming principle which is able to transform one bacteria into some other bacteria, and hence he concluded his experiment. But he could not tell that what is that macromolecule, okay, according to onto the molecular level, what is that macromolecule that is able to convert one particular bacteria into some other bacteria. Now this particular experiment was taken up by 3 other scientists after some time, and these 3 scientists were Avery, McCarty and McLeod. So these are the 3 scientists, Avery, McLeod and McCarty. So these 3 scientists, what they did, they took up the same experiment, okay. So let us say that this is the living mice. So this is the living mice, and along with the living mice, this was the cocktail, right? All those bits and pieces of heat killed bacteria, and all those bits and pieces of heat killed bacteria along with the rough strain, R strain. So now what did they do? They took this particular cocktail, okay. So obviously this cocktail, if you just try to find out that what is it composed of, that it is composed of all those different things. So it had protein, it had nucleic acid, so it had fat, it had sugar, okay. So all the things it had already. So what was their strategy? If you are able to eliminate all other macromolecules and you are able to let one macromolecule survive into the reaction, you'll be able to decipher that which particular macromolecule is able to convert the R strain into the S strain. That when they wanted to survive and when they wanted protein to be active, they added all those other enzymes like nuclease added, they added lipase, and like sugar digesting enzyme like amylase. So all these things they added, but not the protease. So what was happening, that cocktail only had protein intact inside it, all other things were not intact. So what they found out, that into this one particular condition, that the R strain remained R strain, okay. So R strain remained R strain, and along with the mice did not die, actually he lived, okay. So he lived, this was the mice happily living. What actually happened into the next experiment, into when they wanted to save the nucleic acid, so this what happened, that they added protease, they added lipase and sugar digesting enzymes such as amylase. Into this was the only experiment in which the mice actually died. So this is how they actually were able to show that only out of these protein, nucleic acid and fat, nucleic acid was the only macromolecule which was able to transform R strain bacteria into the S strain bacteria, and that's why nucleic acid was the transforming principle and nucleic acid was the genetic material. Okay, so this was the next experiment which actually defined that nucleic acid was the genetic material. Now here comes the next experiment, which also suggested that indeed the DNA is the genetic material, not the protein, known as the Hershey and Chase experiment. The fight was between protein versus DNA. So this was the fight. Now, if somehow you're able to tag protein with something, we're going to tag the protein. So radioactive protein. And if you somehow you're able to tag the DNA, so radioactive DNA. Now, how you're going to get a protein radioactively labeled? So they were working with the bacteriophage. So this is the bacteriophage. So bacteriophage, as the name itself is suggesting, bacteriophage. So phage means virus. So those viruses which are able to act onto the bacterium. Now this particular bacteriophage were grown. Okay, so this was a petri plate, let us say, and this one bacteriophage was grown into sulfur, into an environment into sulfur was radioactive. Okay, and sulfur is present in protein and absent in DNA. On to the other hand, phosphorus is present in DNA and absent in protein. So phosphorus and sulfur can be utilized to tag sulfur can be utilized to tag protein, and phosphorus can be utilized to tag DNA. So if the surface, if the medium is very much rich with the radioactive sulfur, that are all the bacteriophage will become radioactively labeled, but specifically their protein radioactively labeled. If you're going to take some other petri plate, and into this petri plate what you're going to grow, that you're going to grow radioactively labeled DNAs. Phosphorus is radioactive. All the bacteriophage which will grow onto this particular plate will have DNA radioactively labeled. Okay, so sulfur is the blue one, DNA is the green one. So we have already discussed that we had two plates. So this is plate number 1. Here we had a radioactive sulfur, and this is plate number 2, in which we had a radioactive phosphorus. The bacteriophage that is going to come from this plate will have this outer covering. Okay, all outer covering which is composed of protein will be radioactively labeled, and on to the other hand, the bacteriophage that will come from the radioactive phosphorus plate, in them the genetic material inside the bacteriophage is going to be radioactively labeled. Now, so this is, as you can see right here, that we have got two kinds of bacteriophage, one with the outer covering labeled radioactively, and one which will have the DNA labeled. And we know that bacteria, the virus actually insert its genetic material. We do not know that what that genetic material is, whether it is protein and whether it is DNA, but we know that indeed that actually insert its genetic material, and hence it overcomes the machinery of the host. So we allow those bacteriophage to act onto the virus. So obviously genetic material was DNA, so it got inserted inside the bacteria, and all those bacteriophages who had only the radioactive coat, not the radioactive DNA, the coat actually remained outside of the DNA. Now what can be done, that this particular bacteria can be let's say this was the test tube, and inside this test tube these are the bacteria, and the virus actually inserted its genetic material inside the bacterium, and this is protein coat outside the bacterium. Now what you are going to do, we are going to centrifuge this particular test tube, and by this test tube into the supernatant will have all those outer covering of the bacteriophage. Okay, outer covering, and into the palate will have all those cells inside which will have the genetic material which is present inside. We can discard the supernatant, and we can see that into which particular fraction the the DNA turned out to be radioactive. So radioactivity only remained into the those bacteriophages in which the DNA was labeled. So hence we actually can conclude that DNA is the genetic material, not the protein. So the properties of the genetic material. So the number one property of DNA versus virus. Obviously DNA is a double-stranded, the RNA is a single-stranded. The double-stranded has got a deoxyribose sugar, and okay, and all these things are pretty much present on to the next slide. Let's move on to the next slide. Okay, so right here, so let us try to read this particular slide. On to the very first, a molecule that can act as the genetic material must fulfill all the following criteria. So it should be able to generate its replica. Obviously, if you want to make one cell to two cell, the genetic material needs to be divided, and it will divide, so it needs to replicate itself. So this will be the number one property. It should chemically and structurally stable. We do not want our DNA to be very much fragile. We do not want our genetic material to be fragile. So that is why that is why you are going to see that why the RNA is not the genetic material, but the DNA is. Although RNA is the genetic material in some of the viruses, but it is only present in viruses as the genetic material, but not into the cells. And we even do not consider virus to be organism. So why? Because RNA is not stable, while DNA is far more stable as compared to the DNA. It should provide the scope of slow change mutation that is required for the evolution. Okay, so mutation is not bad, but contrary, it is one of the major forces of evolution. But we do not want evolution, or we do not want mutation to happen very rapidly. We want mutation to happen on a steady rate, and steady rate is going to help us out into evolution much better as compared to the that, as you can see, that if mutations are happening and onto very drastic level, that we are going to develop cancers and all other diseases that is happening right now in today's world, where the pollutants or the carcinogen or the chemicals that we are exposed to are enabling us to that mutation rate to go now to accelerate, and that is why we are able to see those diseases in our community, in the population. So mutation rate has to be fairly steady, fairly low. There has to be one particular rate, and it should be able to express itself into the form of Mendelian character. Okay, so the one point that it is supposed to be stable, we can include some of the points that it is supposed to be structurally and chemically stable. So we can say that double helix is helping DNA to become stable. We can say that OH2-OH will make RNA more reactively active. So RNA is, since it has got 2-OH, it is reactive as compared to DNA. Okay, so, and it should provide the scope of slow changes, and like RNA mutate faster than DNA. It should be able to express itself into the Mendelian character. We are done with that. So this was the basic introduction for the history of the DNA or the genetic material. See you into the next video.